So I'm going to talk about the frequency following response. So the frequency following response then is a response that is obtained by delivering sound in the ear, and it is a physiologic response. We only use 3 scalp electrodes, and we are able to measure a response that actually resembles the stimulus very well. So this is the sound wave, and here is the brain wave. And so it gives us tremendous precision in sound processing. And importantly, the person who is getting their response measured does not have to perform a task. So we can get this information from a person without the complication of having them understand and perform a task. So it has been kind of a terminology crisis for the frequency following response. It was first discovered a long time ago, many decades ago, as a response to low frequency pure tones, very simple stimuli. And then we used it the first time for speech in our lab just at around the turn of the century. And as you mentioned, we called it the CABR, the ABR to complex sounds, because we wanted to be able to capture not only the sustained tonal components, but also the transient components in sound that are so important for consonants and vowels. But one of the things that I really regret is this name, because people think of it as, they think of the B in brainstem, and they think of it only as a brainstem response. But really, as I will explain in a little while, it is a response that, even though it is rooted in the brainstem, really reflects the entire brain and reflects what we have learned in our life in sound. So for that reason, we have really come back to the term FFR. And again, there have been so many different words that people have used, and I find it very confusing. So we are just saying at this point, FFR is best. So when we talk about this response, we just use the terminology FFR, and I hope that that will stick. So I wanted to tell you a little bit about myself. This is my mother, and she was a musician, and I grew up in a house where more than one language was spoken. Italian is my first language. And so I was always involved with sound. When I went to graduate school, I first majored in literature, in comparative literature, because I knew some languages and I liked to read. But I eventually discovered the biology of, I started taking biology classes, and I really realized that I liked that very much. And I was very influenced by a book by Lenberg, which was called The Biological Bases of Language. And so that made me realize that I could really study some of the things that I was interested in biologically. I started out looking at the cochlea in the chinchilla, and I was looking at eighth nerve single neuron responses, looking at two-tone suppression in the auditory nerve. And at that time, when I was explaining to my mother what it was that I was doing and how I was spending my time, she was saying, Nina, what are you doing? And I realized that if I couldn't explain to my mother how I was spending my time, I didn't want to be doing that. That made sense to people who were not scientists, and things that I wanted to study, issues that are important in the world. And ultimately, as you know, I'm very interested in using the work that we do to influence education and medicine. So then I started recording from single neurons in the auditory cortex, and I was using the rabbit as a model. And if you play a sound and you record from a single neuron, you see the neuron will respond. Now, if you teach the animal that the sound has meaning, so here we have trumpet means more carrots. So the animal learns that something will happen when the sound occurs. The animal is the same, the sound is the same, but the brain has changed. So this made a very big impression on me, because this showed me firsthand in single neurons that the brain has the ability to change from its experience with sound. So I think I have spent my whole career studying how our life in sound changes our brain. And of course, I am most interested in using and applying what we know from the animals to humans. And if you look at, snapshot is from the home page of our website, Brain Volts, that I hope you will spend a lot of time on. But you will see that we study many things: music, concussion, bilingualism, aging, listening and noise, reading, autism, neuroeducation. We do technological work. And you might wonder, what are they doing at Brain Volts? But really, the overarching idea is sound and the brain. Sound is important for all of these activities. And so, you know, sound is wonderful. Sound is vast. It is a big part of our lives. And as you can see, it encompasses all of these different areas. So in the end, I'm very happy using the frequency following response. And so let me tell you a little bit more about it. So first, let me start with sound. Sound, you know, we live in a visually dominated world. And if you think about a visual object, you can name the ingredients very well, because you can see them and they're right in front of you. So an object will have a texture, a size, a color. You can describe all of these things very concretely. Now, sound also has ingredients, and it has many, many, many ingredients, but they are not as obvious because sound is invisible. And sound is an invisible, powerful, powerful force. You know, many of the world's most powerful forces are invisible, like gravity. But they are a part of us. And so here are some of the ingredients in sound that we hear. Now, the way I like to think of sound in the brain is that these ingredients are delivered to the brain, and then the brain makes sense of these sounds by processing each one of these different ingredients in its own way. So we are not talking about a volume knob effect where all the sounds and all the ingredients are processed in the same way. They are, in fact, processed distinctly. Each ingredient is processed distinctly based on our inherent biology and on our life in sound. So I like to use this mixing board analogy to think about how the brain processes the different ingredients in sound. So we can actually measure the brain's response to these different ingredients. So again, here's the frequency following response. We deliver sound into the ear, and using just 3 scalp electrodes, we can measure a response that looks like and even sounds like, so here, so you can hear that the sound wave and the brain wave actually sound like each other. And here are the different faders on the mixing board that can tell us how good a job the brain is doing processing these different ingredients. So I'm going to talk about each one of these ingredients one by one. We can analyze the response that we get, the brain wave, by doing a Fourier analysis. And in the frequency domain, we can measure the strength of the fundamental frequency and also the strength of the harmonics. We can look at the different timing components. You can see there's so many things that we can measure in time. And especially for consonants, timing is tremendously important. So we can look at how does the brain process timing in quiet, and how does it process timing in noise? This is something that I know you are very interested in. And we can look at how accurate the response is by computing a stimulus to response correlation. Now, if you can imagine, the response and the stimulus are so similar that it is actually meaningful to correlate one with the other. And the better the correlation is, the closer to 1, the better the response looks like the stimulus. And so we can compute a stimulus to response correlation. It's a very simple computation. We can also, before I go to stability, we can also compute the response to response correlation. So for example, we might want to correlate the response in quiet to the response in noise and see how much the noise disrupts the correlation. We can look at how stable the response is from trial to trial. This will turn out to be a very important component. And we can look at phase. So phase is, again, another form of timing. And if we look at the timing components in a consonant, for example, we can also compare the brain's response to the phase differences in, say, a ga versus a ba. And the redder this is, the larger the timing difference. So it is telling us that the brain has computed a very good distinction between ga and ba, because we can see that there's a large phase difference or a large timing difference. Importantly, the FFR is different in every person in the same way as our written signature is different. We are all writing letters, but our signatures are different. So here is one participant and here is a second participant. And if you measure their response 4 years later, you see they pretty much look the same. So that's really important. And if there are tiny differences, you can really measure them and they are significant. Interestingly, also, there are differences. So as we look at norms for the frequency following response, there are differences between males and females in some of these different components of sound. So some components are the same in males and females, and some are different. So I will ask you to take a look at a paper in hearing research that looks at sex differences, so that you can look at the normative data and see that there are differences in males and females. So the frequency following response is very powerful to me, because number one, it reflects how good a job the brain does at processing different sound ingredients, and it reflects our experience in sound. We also, it's actually a lot of fun, and it's interesting and informative to listen to the FFR. So in the same way, we are all familiar right now, I can be playing an instrument or talking to you, and the sound wave is taken by a microphone, and it is transformed into electricity that is then played out of a speaker. Now, we can listen to the sound and measure the electricity with scalp electrodes. And again, we can play the brain's response. And I just want to play a few examples of the frequency following response to you. So here is the stimulus. And here is the brain's response. So we have a lot to work with. And I wanted to have you listen to 3 healthy brains, to have you hear how, these are all healthy brains, but we all hear the world a little bit differently. So here is one brain recording, listening to the Beatles song. Here's another brain. And here's a third brain. So I hope that you can hear that even though the melody is the same, each brain processes the sound differently, which we can see in the response, and we can also hear it. So taken together, the FFR is objective. It is very well vetted. It is mobile. We can measure it anywhere. It is fast. It is repeatable. And it is uniform in that you can measure the same response in an infant, or in an older person, or in an experimental animal. And so this enables us, if you think clinically as an audiologist, you use very different tests for a baby and for an adult and for an older person. How wonderful to use the same measure across the lifespan. I invite you, please, on our website, which we really, our website is a labor of love. It's called Brain Volts. We have created a little demonstration of our biological approach. It is on the home page, and it's maybe 3 minutes long. And I encourage you to take a few minutes and to watch and listen to that. So how does the brain make sense of sound? So sound, of course, has to enter the brain, and we have an afferent pathway And of course, we have an efferent pathway, which influences our perception of sound. And our perception of sound is very much influenced by what we think, how we move, our other senses, and how we feel. So our hearing system is really vast. And I think that that is really something that that is often overlooked. You know, people think of the hearing system as involving only the classic auditory pathway, but in fact, the auditory pathway is very much informed by the rest of the brain. So, for example, if you listen to this sentence, it might be very hard to understand. Once you know what the sentence is,
the juice of lemons makes fine punch.
Now, I want you to hear the first sentence again. And I hope you have experienced that what we know about sound influences how we hear it. This is just one of many, many, many examples. So we have, again, tried to consolidate this thinking and put it all in one place. We wrote a paper for Trends in Cognitive Science that talks about the cognitive, sensory, motor, and reward pathways that are a part of our vast hearing system. So this is how we think, move, sense, and feel. So, you know, the classic pathway is this afferent pathway from ear to brain. And of course, we have an efferent system, which is even more massive than the pathways going upstairs. Importantly, it is informed by how we feel, how we think, how we move, and how we sense the world. So let's think about the auditory midbrain, which is really where the frequency following response, the FFR, is largely a midbrain response, but it is not merely a midbrain response, because the auditory midbrain is a hub. It is informed by the afferent pathway, and it is informed by the efferent pathway, and it is informed by how we sense, think, and feel, our sensory, motor, and reward system. So the midbrain is really the hub of our hearing system. So it becomes a very, very, very powerful, we can really think of it as providing a snapshot of auditory processing. So let's think about this just a little bit more theoretically. We have our afferent pathway, which is primarily an automatic default system. It doesn't change that much moment to moment, but and we have our efferent system, which is especially malleable, and importantly, with experience, as we learn to make sound to meaning connections. For example, we learn the sounds in our own language. Our brain automatically makes these responses. Our afferent system changes to a new default system. So, for example, some of the work that was so influential to me was some work that Ravi Krishnan did a number of years ago, where he measured FFRs in tonal speakers, so people of tonal languages, and he found that speakers of tonal languages had FFRs to tone changes that were stronger than people who did not speak a tonal language. And importantly, they measured these responses while the participants were sleeping. So this had really become automatic. This had become their default system because of the many experiences that they had in their lives making sound to meaning connections with these tonal sounds. So the sounds of our lives very much shape our brain. And we can be auditory experts. We can have difficulties. And so I think of sound as being both an ally and an enemy of brain health. So now I would like to talk more about the specifics of the frequency following response. We can take these ingredients and think in terms of pitch and timing and timbre. A very important aspect of pitch is found in the fundamental frequency. Timbre is represented by the harmonics, and of course, there is timing everywhere in sound. So if you take any sound, let's see, a musical instrument or the word taco, we can see that there are many, many timing ingredients in the sound, not only the onset and the offset, but all these different components, lots of time information. We can look at a part of the response and find the fundamental frequency inside. And then if we now zoom in to the fundamental frequency, we can see these different components here. And what they are is the timbre or the harmonics, the fine structure, and they are all present in any sound. So with the frequency following response, here is a stimulus, here's a sound, and here is the FFR. You can see the timing, different timing ingredients. We can look at the pitch or the fundamental frequency. And we can look at the timbre or the harmonics, both in the time domain as well as the frequency domain. Okay, so I hope that you can see how we are able to capture such a rich amount of information of how the brain processes sound. This is so much more than any other auditory evoked potential is essentially an onset response. It does not give you all of this information about the different sound ingredients. So the FFR is very powerful indeed. So if we think about what stimuli we might want to use to elicit the FFR, in order to maximize the transient responses, you need a stimulus that has a nice strong onset and bursts in amplitude. So here, for example, is a cello note that gives you this very nice response at the onset. Here is a baby crying. And here is a piano melody. And again, you can really see the onsets and the transients very well. To maximize the sustained responses, if we use kind of a lower pitch sound, you can have very strong phase locking to the fundamental and the harmonics. So again, you can see here is the stimulus and you can see the phase locking, and here is the response. And again, you can see the phase locking reflected in the response. So if we think about speech sounds, like a speech sound like da, the consonant is primarily high frequency and it has diffuse energy. It's a little bit like a click. And the vowel has more low frequency bands and discrete energy bands, and it's periodic. And it has more tone-like components. But, you know, in the end, we are really interested in sounds that are important to us, like speech and music. So we have our own sound library where, you know, we have sounds that we have carefully created. So we have da and ba, ga, ya, ya that goes ya and ya. So with a pitch trajectory, we have da and noise. These sounds are freely available. If anybody would like to have our stimuli, you just need to send me an email and I will send you whatever sounds you desire. Now let's look at the response. So let's look at timing. This is the response to our 40 millisecond da. You can see that there are different timing components happening at different latencies. And you can measure da in quiet and in noise. We can take the response from of the da stimulus, and again, you know, what we're doing is an FFT, so frequency, a fast Fourier transform. And so here's the fundamental frequency, and here are the harmonics. And so again, here is the FFT of the FFR. So here is the fundamental frequency, and here are the peaks of the harmonics. And this is the noise floor. So you can see that the fundamental frequency and the harmonics are well above the noise floor. So importantly, we can analyze, you can analyze the entire da stimulus, but you can also just analyze the response to the ah component, or just the response to the d. And again, you have a wealth of information from one response from the brain. You have a tremendous amount of information about how the brain processes consonants and vowels, transient and sustained portions of sound. So let's look at stimulus to response correlations. So here is the stimulus da, and here is the FFR. And again, the fact that the response and the stimulus look so similar allow us to make these correlations. So if you correlate one with the other, you'll basically get a correlation that is closer or farther away from one. A perfect correlation is one. So here is the stimulus in gray, and in black you see the FFR. And so you correlate one to the other, and you see, all right, for this person, there is a stimulus to response correlation of 0.6. So it gives you a very objective, easy to compute metric. You can also look at response to response correlation. So on the one hand, we can compare the response to the stimulus, but you also might want to compare one response to another. So for example, you might want to compare a person's response in quiet to the FFR in that same person in noise. So if you do a quiet to noise response to response correlation, then again, you will see how much of an impact the noise had in affecting the or degrading the brain's response to the sound in quiet. We can look at pitch tracking. So when I make when I'm speaking with a sentence and I'm asking a question, my voice goes up, or if I'm making a statement, it goes down. And if we have a simple trajectory of a stimulus that goes up, like I'm asking a question, we can look at the frequency following response and follow it over time and see how good a job the brain does at tracking the pitch of the stimulus. And you can see that this correlation is very good. We find actually that some children on the autism spectrum have very poor pitch tracking because they often have difficulty, in fact, understanding if you have asked a question or if you are making a statement. So this happens to be a child on the autism spectrum with a very low pitch tracking. Response consistency is tremendously important. So if we present the da over and over again, which is what we do when we record the response, of course, it's an average response, we want to see how consistent is the response from trial to trial. This is a very inconsistent response. You can see how jittered the response is from trial to trial, whereas this one is a very consistent response. And one of the reasons this is important is that in language disorders, we find that the response consistency or the stability of the response is very poor sometimes. And you can imagine how it would be a problem in language, learning language and learning to read, if every time you hear a sound, your brain processes it differently. If it's not a stable response, how are you to learn? So response consistency is a very important computation, and it is one in which you basically compute how consistent is the response from trial to trial. And again, you can look at how consistent is the response to the consonant in a d, da. How consistent is the response to the vowel? So again, this can be very important. And again, we find that especially in response to the consonant, this is something that kids with language disorders really have difficulty in terms of the stability of their response or consistency. I'm using the terms stability and consistency synonymously. Now we can look at phase. So if we look at the difference between a ga and a ba, we can create a stimulus that just is different according to the formant frequencies of the sound. Otherwise, the stimuli are the same. So these are sounds that we can manufacture ourselves very carefully. And we can see that if the brain is computing this timing difference between ba and ga, again, we can see how good a job the brain does telling these two sounds apart. And again, this is very objective, and it's something that can easily be done. You don't have to be picking any peaks or making subjective decisions. You're really just looking at the response to timing. Now, we're getting to polarity. So when we present a sound like ah, we can present it with 0 degrees timing or 100 degrees out of phase with each other. And this is the way we often record the FFR. We will present a condensation and a rarefaction stimulus, right? So we will use alternating polarities. But what is interesting is that we can use this, even though this sounds the same, but the brain can tell these apart. If you add the two polarities, you get a response, and this is usually what is done with alternating polarity. You add the polarities, and it maximizes the fundamental frequency, whereas if you subtract the response to the two polarities, you are biasing the response to the higher frequencies. So this emphasizes the fundamental frequency, and the subtraction emphasizes the harmonics. So again, imagine how much information you have in a single response. You can have a single response to a DA stimulus. It was collected with the two polarities, and you can take this response and add the response to the two polarities, or subtract them, and from the same response, provided that you have saved the added response and the subtracted response separately, you can compute responses that emphasize the lower frequencies or the higher frequencies. So from one response, you have a lot of information. And the brain actually listens when we're listening to sound. We're listening to information in a single polarity, and we can also look at the brain's response to to a single polarity and get both the low and the high frequencies at the same time. So here are some of the things that are necessary for recording an FFR. Any kind of ABR system will do. That doesn't have to be anything in particular. We like to use this DA stimulus at 80 dB SPL because we have a lot of norms and we understand it very well. Ideally, we would like to be able to save the response to individual sweeps in the average. And importantly, you need the ability to perform offline analyses, FFTs and correlations. We do a lot of this in MATLAB. Norms, we have norms that are published, or you can create your own for your stimuli. So for our norms, for the short DA, we have a normative paper, and for the long DA, we have another normative paper. But I encourage you, again, this is a panel from the homepage of our website. If you press this panel, you click on it, you will see a list of our parametric studies that will give you norms for the short and the longer DA stimuli across age and also sex. So To present the stimulus, you really need precise timing in triggering the recording system. So I think many people are frustrated because they use recording devices that are used to record cortical potentials. And most of, any cortical potential, you're looking at responses that are changing over tens of milliseconds. But for the FFR, you are looking at responses where tenths of milliseconds are meaningful. So you cannot have any jitter in the triggering of the recording system. In other words, between the delivery of the stimulus and the recording of the response. It's nice to have continuous and simultaneous masking if you want to use it. We use usually a pretty fast presentation rate. We record the data in alternating polarity, and we save the responses to each polarity. We use well-shielded insert earphones, and it's also important, and as audiologists you will know this very well, but many other people do not know this, that it is really important to place the earphone in perfectly, because if you don't, you will not have a valid response. If you would like, you can have multiple stimuli presented in the same sequence. And the electrodes, you can use silver chloride electrodes. Actually, we have been moving to disposable electrodes, and they seem to be working quite well. To record the response, you need a sampling rate of at least 10 kilohertz, because again, you are measuring responses that are very, very fast. So you need a fast sampling rate. Differential amplification helps you get rid of noise that is extraneous to the response to the stimulus. We often use this filter bandpass, especially offline. You need amplitude accuracy as well. You need to have a mechanism to export the raw data for analysis with other software. Obviously, you need to do online averaging, online artifact rejection. The ability to save individual sweeps is ideal. And the ability to view an artifact reject on an average at a different filter setting than the file being saved. So for example, you might collect the data with your filters at 70 to 2000 hertz, but you might view the average while you're collecting it at a different band pass, so that it's easier to interpret just while you're collecting online, so that you can be sure that as you are collecting good data. And in the same way, we find it very important to have an ongoing display of the EEG that you're collecting. So, importantly, how do we use the FFR? And we use it in education and medicine. In communication, it really is a way to give us information about a person's language ability, how well they can hear a noise, and it gives us information about their brain health. We've been doing a lot of work these days with athletes who have had concussion, and it turns out that the hearing brain can really give us a lot of information about brain health. We can look at auditory system plasticity on multiple time scales. In the moment, so this is the effective attention, if you will. The effect of short-term training. So what is the effect of using a hearing aid for 2 or 3 weeks? And what about lifelong experience? The languages we speak, the music we make. It's extremely an important way, powerful to look at therapeutic intervention, because the most powerful way of looking at the response is within a subject. So using a subject as their own control. So imagine if you measure the FFR in a person and then they do some kind of therapy, and then you measure the same person again after the therapy is completed. So, we have various applications in music, in the classroom, in learning, in audiology, and for brain injury, the auditory system and the FFR can really give us an objective index of the health of the brain. When we think about auditory processing, auditory processing really involves, listening and noise, language, auditory processing disorders. We have these different disciplines, which makes looking at auditory processing difficult sometimes, because there are the speech pathologists, and then there are the audiologists, and then there are the learning specialists and the physical therapists. But in fact, as I hope I have convinced you, auditory processing consists of all these components. And I like to say that nature does not respect disciplines. So the brain processes all of these different aspects of sound. So I think we have to be looking broadly. If you can imagine a sample report, so here is your stimulus da, and imagine how well the person that you are measuring the response from is doing in terms of timing, stability, and harmonics. Takes about 20 minutes. By the way, one of the things that we like to do is we record an ABR to a click at the beginning, and then we record the FFR, and then we record the brain response to the click again at the end, so that we know that the earpiece hasn't shifted. And we also have the response to the click. So, we have this response that the timing will tell us how quickly and accurately the brain processes sound. The stability will tell us how consistently the brain responds to sound. And in the frequency domain, we can see how well the brain processes pitch and timbre or harmonic cues. And we can think about, so this might be a sample report. So here is our template response to da, and here is our response of the patient. You can see that there is delayed timing. And you can see that if these are the norms for stability, our patient is very low. So he has a very poor stability or consistency. And then if you look at the processing of the fundamental frequency, this particular patient is perfectly within the norms. But you can see that the harmonic processing is reduced. So here you have somebody who has delayed timing, poor stability, and disruption of the harmonics. And that to us is very much a signature that is associated with auditory processing and language disorders. So if we look at these different components, timing is very important for phonetic awareness, for the brain to become more stable. Training attention to meaningful sounds is an important strategy. And the fundamental frequency is very, very important in picking out stimuli in noise. That's a very important cue. And by the way, one of the things that we have looked at in the past that you might be interested in is we have looked at assistive listening devices in the classroom. And after kids have been wearing these devices for a year, their brain responses become more stable. This is a paper that we published in PNAS, in the Proceedings of the National Academy of Sciences. The first author was Jane Hornicle, and I encourage you to have a look at that if you're interested in assistive listening devices and how that might change the stability of the response over time. So these are the various strategies that we can use, and of course there are many more. I do want to say that language disorders and listening and noise have some overlap in terms of the ingredients that are affected by FFR recording, but they are also distinct. So I would like to just give you an example of these two. So if we're looking at language disorders, if you present da in noise and we look at timing, the FM sweeps, so in other words, the timing across the formant transition, the harmonics, and how consistent the response is, these are all ingredients that are very important for language. That again, you can look at the details in these publications if you would like. I really do think that in the future we ought to be able, in the same way as we do newborn hearing screening for hearing loss, we should be able to screen babies to see if they are at risk for language and learning problems. In terms of hearing in noise, these are the components that are important: the fundamental frequency, timing in noise, and the distinction of speech stimuli. And I think we are all familiar with people who have the same audiogram, but one person has an easy time in noise and the other person has a good time in noise. In other words, some people have good hearing and noise and some people have poor hearing and noise, even though they have the same audiogram. And what we can see is that the person who has the poor hearing and noise has a frequency following response that is not as good, you can just see right away, as the person who has the identical audiogram that has a very nice frequency following response. We can also see that experience really matters. So here is a musician with a hearing loss, and the musician actually has better hearing and noise than the non-musician, even though his audiogram is worse. So again, this is a testament to the importance of the brain in shaping how we respond to sound. And again, if you look at the frequency following response, you can see that the response in quiet is this response with a large fundamental frequency. And in the non-musician who has difficulty in noise, you can see that in noise, the fundamental frequency is much reduced. But in the musician, you see that the fundamental frequency is just as good in noise as in quiet. So this gives you a lot of information to work with. Auditory working memory is also better based on a person's experience, not their audiogram. So I think it's very, very important to be moving towards using FFRs for hearing aids. So if, for example, if you have a hearing aid and you have a response that looks like this, so this is the FFR in the time domain and in the frequency domain, and a person is saying these hearing aids aren't working well anymore, then you give them a different hearing aid or a different setting, and you can see the same person now has faster, larger responses, and importantly, a larger fundamental frequency. And so here is now the person with the new hearing aid saying, I can't remember the last time I heard so well in a restaurant. So again, I really encourage, to me, this is a really important open field for audiologists to be able to say, to use the biology to help us understand and choose what is the best hearing aid and the best hearing aid setting for a particular person, because every brain is different. Samira Anderson is doing a lot of work in this area. She's at the University of Maryland. And if you look at the unaided response here compared to the aided response, she can really show with the FFR how the amplification boosts neural synchrony. So you can imagine, this is not something that is currently available, but one can envision if you had automated fitting where you were rapidly collecting FFRs to many different algorithms. And then you see for a particular person, oh, this person gave me the best response to this particular algorithm. So this is the hearing aid, this is the algorithm that I'm going to use for this person. So this is the kind of possibilities that I envision. So of course, we can use different devices, software training, and I think it's very important to have an objective biological metric to look at the effects of training. And again, do have a look at this particular article that has looked at auditory training in older adults and has found that the FFR becomes more precise, hearing a noise gets better, and auditory memory gets better with software-based training. Music is always a very good strategy for strengthening your brain. And assistive listening devices. Again, as I'd mentioned before, we have found that brain changes in the response, the FFR, after children have worn assistive listening devices for a year in the classroom, that has helped them learn what to pay attention to. It has really enabled them to focus their attention on the meaningful sounds. So our vision in assessing hearing is, we ask these three questions. Is sound getting in? How well does the listener use sound? And how well is sound processed by the brain? Well, we use conventional audiometry to answer this question. How well does the listener use sound? I think it's important to measure speech and noise ability, auditory working memory, and attention skills. And these are some of the tools that we like to use, but you can choose whatever you like. But I think that it is important to know how well a listener uses sound in the world. And then the big question is, how well is sound processed by the brain? And this is where the FFR can give you a tremendous amount of objective information. We have a whole book on the frequency following response, which I encourage you to see if you can get a hold of. I really encourage you to look at this brand new article in Hearing Research. Krisman is the main author, Krisman and Kraus. And this really gives you the detail in case if you missed something in the presentation today. How we analyze and record the FFR is all in this paper. So I hope that you will use it and you will share it. And if you go to our website and look at our technologies, you will see, again, a lot of our norms that might be helpful to you. I encourage you to please visit our website, and especially we now have a website tour that I hope you will take. If you go to the top of the website, you can see tour the website. And again, that's a 3-minute tour that will tell you what's on the website. And one of the things that is on there is under publications, there is a link for clinicians where we have all of the hearing journal articles that we have written all in one place. And so I hope that you will enjoy those. These are the current members in the lab. And of course, our work is based on all of the people who have worked in the lab over the years. And I think that's what I have for you today.